We've got one team left, one finalist team left, Team yOIL. And presenting for Team yOIL is Sarah Farr, who's working towards her undergraduate degree in plant biology from the University of Calgary in her native Canada, our neighbors. Hi there, Sarah. How you doing today?
Hi, I'm doing well, thank you. I'm really excited to be here today.
Good. Well, we're excited to have you. Go ahead and take it away.
All right. So my name is Sarah, and I'm going to be presenting on behalf of Team yOIL today. Canola is one of Canada's largest industries, worth $27 billion. But stakeholders across the industry lose $730 million every year due to how we manage two wicked problems. And these two problems are the green seed problem and the sclerotinia problem. The green seed problem occurs when inclement weather causes seeds to contain the green chlorophyll pigment at the time of harvest. These green seeds are more difficult to process, so they sell for a lower price, and this means that farmers lose $150 million every single year. Sclerotinia, which is a fungal disease, causes stem rot in canola crops and results in more than 50% harvest loss when it infects the crops. So farmers end up losing $580 million every year from this damaging fungus. Harnessing the power of protein engineering, our goal is to create a sustainable and circular solution to these problems. So the canola oil value chain follows this general pattern. First, canola plants are grown, the seeds are harvested, and then they're pressed into a green oil, which is then processed to yield a consumer-ready yellow oil. But because of the sclerotinia and green seed problems, there are excessive input costs and waste involved in this production process. To deal with sclerotinia, farmers spend on average $21,000 on fungicides. But these fungicides only prevent and don't actually treat the infection, which means that farmers are taking a gamble every time they decide to apply it to their crops. The broad range effects of the fungicide are also detrimental to the surrounding ecosystem. In order to get rid of the green chlorophyll pigment, the industry currently uses acid-activated clays, but the processing conditions are costly and the yield is very low. Worst of all, the clays are single-use and cause a lot of damage to the environment upon disposal. The process stream also has two additional waste products. The first is canola meal, which is the leftover seed mush from pressing out the oil. It's mixed with the acid-activated clays and sold as animal feed with damaging effects on the surrounding ecosystem. The second is gum, which is a thick oil byproduct that's removed from the pressed oil during processing. The lack of end-use market means that the gums are just burned off or disposed of by a third-party company. So these excess input costs and waste make for a really unsustainable system. Inspired by the three tenets of the circular economy, our team looked to using cleaner biological alternatives to circularize canola oil processing. And we've designed a system that tackles both of these problems together by using the green seed problem to solve the sclerotinia problem. So to do this, we turned to protein engineering. We developed a new chlorophyll extraction system that uses a protein which can be emulsified in the oil to naturally collect the chlorophyll pigment. And the advantage of this solution is that it's reusable, meaning that after the chlorophyll pigment is collected and removed from the oil, it can be used for something else. And that's where the sclerotinia problem comes in. We actually found that the chlorophyll pigment can be degraded into thiophormide, which is an anti-fungal agent that inhibits the growth of the sclerotinia fungus. But what about the gums and the canola meal that were unused waste products in the previous production stream? Turns out we found a use for both of those products. So the gums that are collected from the canola oil can actually be used as a surfactant, as a part of the biological chlorophyll extraction process. In addition, the pure canola meal can be used as a feedstock material for the bacteria that produce the protein, and can also be sold as a natural fertilizer as well, with no damaging effects on the environment. Together, this results in a system that benefits all stakeholders whilst cutting out excess waste and providing a sustainable circular solution to multiple problems in the canola industry. And the best part is, this isn't just a theory anymore. So our project is 1 year in the making, with our novel advancements in genetic engineering winning us a second place prize amongst 300 teams at the International Genetically Engineered Machine Competition, beating out teams from Oxford and MIT. We've spent the last year developing a proof of concept for our protein emulsion system, which shows marked improvements from the previous clay system. Our first improvement was reducing the energy costs. The clays require high temperatures for efficient processing, but in comparison, our chlorophyll removal system is designed to operate at room temperature, and it removes twice as much chlorophyll whilst doing so. In addition, our system was designed to minimize the oil loss. So our method retains 92% of oil, whereas acid-activated clays lose over 20% in the manufacturing process. We've also been able to demonstrate that thiophormide works as an antifungal agent, with increasing concentrations showing increased inhibition of the sclerotinia fungus growth. But this isn't the end of the road for us just yet. With our award-winning technology under our belt, we look towards establishing ourselves as a business on the canola market. Due to the high R&D costs and timeline associated with fully developing a fungicide for commercial use, our team first decided to focus on bringing the chlorophyll removal system to market. So, we first started by looking at the processing cost for canola oil. Oil producers usually purchase acid-activated clays at a fixed cost. In comparison, the biological nature of our system allows for an exponential decrease in the cost required to produce our protein as the total processing volume increases. As a result, we realized that we wouldn't be able to outcompete clays with cost efficiency with protein production volumes lower than 40,000 tons annually. But our target is to partner with an oil producer that produces a total volume above this threshold, so our proteins are more profitable. Knowing that a minimum process volume of $40,000 was necessary, we reached out to Richardson Oilseed, a large local oil processing company. But we learned from them that entering the market through large-scale processing wouldn't actually be so simple. Upon hearing about our design, Richardson Oilseed first told us that it was crucial that our technology integrates with existing equipment at manufacturing facilities for any oil producer to even consider a partnership. Additionally, we learned that larger scale oil manufacturers are a little bit more resistant to implementing new technology into their already existing tried and true manufacturing pipelines. So with these three challenges in mind, we decided that the sweet spot to target would be to partner with smaller but still industrial scale oil producers. Although each oil producer that's small produces only approximately 18,000 tons of oil per year, if we partner with three, that would push us above the 40,000 ton per year oil production threshold, allowing our proteins to be sold at a competitive price to the acid-activated clays. We also made it a point to design our protein production process and emulsion system to require no additional infrastructure whatsoever at existing oil facilities. Last but not least, our partnership with small-scale oil producers would allow us to validate our model on a relevant industrial scale, and it would allow us to optimize the process conditions and reputation that we need to later integrate our system with larger oil producers. With these three objectives in mind, our role in this partnership would be to produce this protein, sell the protein to the oil producer for use, and then buy back a portion of the canola meal produced in the oil production process, which then we can sell as a fertilizer for profit. With this system, each small-scale oil producer actually saves $4.8 million per year. Their bleaching process cost goes down from $707 per ton of oil with the clay system down to a whopping $438 per ton of oil. This is a 38% decrease in their overall bleaching expense. On our end, this results in a 7.17% operational profit on an annual basis. As we expand our system to more oil producers, this operational profit is expected to exponentially increase. Based on published recombinant protein production models, we estimate our initial startup cost to be approximately $2.2 million distributed over 2 years of construction. These costs are distributed over renting space, installing necessary equipment, controls, electrical systems, and contingency costs in case of emergency. Because our system is designed to integrate into existing oil production facilities, the startup cost falls on us. And although $2.2 million might seem like a lot at first, we've predicted the cash flow of our model over the next 5 years, where our break-even point would occur near the first quarter of our 4th year. By the 5th year, we would reach a post-tax profit of $750,000 per year. As our system becomes more established and net profits increase, we plan on reinvesting this excess capital into research and development for the full characterization of thiophormide as a fungicide, so we can make steps towards fully circularizing our system. And also, we want to look towards scaling our chlorophyll removal process to various edible oils, such as soybeans. So with these plans in place, you may be asking us, where are we currently? So far, our team has been able to validate our technology in the lab, and with the support of various programs and organizations, we've secured our first prize money from a pitch competition, bringing us to where we are today. In the future, our goals are to optimize our procedures in the laboratory and then turn to partnering with oil production facilities to test our tech. yOIL started out as a modest research project, but has grown into something else entirely. The five of us spent the last year attending canola trade shows, talking to farmers, and rooting ourselves in the industry, and we are proud to have found a home in this innovative corner of Alberta. With the future in our hands, we must work together to responsibly change our biggest industries. By reducing waste, minimizing environmental harm, and closing the production cycle, we can create a brighter future. Let yOIL be the change you wish to see. I'd like to thank our sponsors that have helped us on this journey, and I will now be accepting questions.
Excellent work, Sarah. Thank you so much. We're going to chat for a minute or two while the judges chat, and then the judges will give you about 10 minutes of Q&A. So Sarah, incredible. That was phenomenal. I'm curious, what inspired you and your team in the canola oil space?
My whole team is based in Alberta, so all of us, we're proud Albertans, we're proud Canadians, and we know that agriculture and the canola industry is really, really big here. So we wanted to make an impact on a local problem, something that we can help our friends and family with. So that's how we settled on looking into the canola industry and trying to tackle one of the biggest issues that they have, which is the green seed problem.
Incredible. You know, I know a bit about canola oil. I know people with autoimmune disease, it's inflammatory for them. And I'm wondering if with reducing the bleaching process, if that would be something that people with autoimmune disease can more easily digest. It's interesting.
There's a lot of research that's being done on improving like the components of canola oil itself. And that's also something that yOIL is really interested in looking into in the future. And also as a plant biology student myself, it's something that is, I guess, on the table for pursuing as a part of my studies in the future as well.
Well, good, thank you. On that note, you're still an undergrad. What is your plan for the future?
Actually, I just finished my undergrad this semester, and I have the plans to start my master's in plant biotechnology and research in the fall. So that's going to be really exciting. I hope to pursue some components of this project as well as a part of my master's program.
Incredible. Are you staying in Canada?
Yes, I'm staying in Calgary, Alberta.
Fantastic. My grandparents are Canadian, so special affiliation.
Yeah,
wonderful. Judges, all set? Great. Good. Well, take it away.
I'll jump in since I have a question with the thinking underlying your solution. So I applaud you on a really interesting technological fix to a current industry that is wasteful. My question is around, have you thought about why we're in this problem in the first place, in the sense of industrialized agriculture with monocrops being the way that we do things, with canola being emblematic of that, soybeans you mentioned is another example. There's a huge amount of side effects, including the need for pesticides, the need for a lot of tech fixes in order to prop up a system that is fundamentally problematic from an ecological standpoint. And there are other approaches to farming that are showing that the use of perennials, the use of interspersing crops, so that the whole system from an ecological perspective is more balanced. You know, there's alternative ways to do things. So I'm just curious whether it seems to me that your approach takes as for granted that the current system of industrialized agriculture is here to stay. And I'm wondering if you've thought of alternative futures as being part of your vision of a future that you can play a part in.
Yeah, so you're definitely right. Like there are a lot of different ways that problems in the agriculture industry can be tackled. But when it comes to the canola industry and the green seed and sclerotinia problems in particular, we actually look to tackle these problems because these are problems that aren't easily mitigated by just doing crop rotation or introducing other species into the field and things like that. The reason for that is the green seed problem is dependent entirely on the weather that year and doesn't have anything to do with any of the surrounding organisms. So if you plant your canola and then you have an early frost that year, that's game over. You have a lot of chlorophyll in your seeds, and it's not really something that can be mitigated by like altering the ecological environment surrounding the crops. And also when it comes to the sclerotinia problem, the spores for the sclerotinia fungus are very, very resilient, more so than a lot of other fungi. So other fungus-related problems can be solved via crop rotation, but this particular fungus is very persistent and it can't be. That's why we thought we might look towards a more technical way to tackle this problem.
Interesting, thank you.
Yeah. I would love to know more about this protein that you've developed, how it was developed, and it seems like it's not a naturally occurring protein.
It actually is a naturally occurring protein. So a certain group of ferns contain this protein that transiently binds to chlorophyll in the cytoplasm of the cell, in the cell soup, I guess you could say. So this protein does occur in nature, and it was characterized as a part of a basic science research project that we found through literature review. But on our end, we've made some modifications for it to suit our system better. So we've modified some residues in the protein to make it better bind the chlorophyll. So yes, it is naturally occurring, but we've just modified it for our purposes.
Is there any danger of that protein getting out into the wild with those modifications, or certainly in scale in a place where it might naturally not have occurred?
So one of the biggest benefits of our system is that the protein production occurs in one facility and it's all contained in that facility. And the only thing that leaves the facility is the protein and not the genetically modified organism that produces it. So we have, I guess, designed our system to contain the bacteria so that we won't release it into the wild just to mitigate any possible environmental effects. And the protein itself, because it's actually purified out of the oil before it's sold to consumers, it doesn't end up in the final product as well.
If the protein is that much better at processing chlorophyll, could it be used in other applications as well?
Yes, actually, one of the biggest, I guess, future directions that we want to look into is the use of this system for algal biofuels. So the presence of chlorophyll is one of the biggest issues in biofuel processing as well. So if we could also adapt this system to remove chlorophyll from biofuels, it would really, really help the fuel industry, which is another big industry here in Alberta. So it's great for us.
Great, thank you.
Yeah, it's really, really excellent. I find all your research and obviously you've already been working on this for a couple of years and getting finding out more and more information about it. And it's great to hear that it has other applications. And also, I must comment, I like your jean jacket mafia sort of picture there, where you guys all be clothed appropriately, or this must be, you're like a team. It's great. I really appreciate
Thank you.
the work and that you've really explained everything very clearly. So to me, it sounds like such an excellent use of the science and how you're able to apply it to an industry that you've identified that it has such a deficiency in its present model. So it's great stuff.
Thanks.
Sarah, I'm interested. Oh, by the way, I also think great presentation, very polished, and I can see why you've been successful. Could you tell us a little bit about the origin story of this idea? Where did it all come from?
So this project started out with an undergraduate team competing in the International Genetically Engineered Machine Competition. It's like a big synthetic biology competition that happens annually in Boston. But my team that was based at the University of Calgary, we looked to use biology to solve problems that existed in the real world. And being a plant biology major myself, I talked to my PI and some other people involved in the industry and was kind of feeling around for any issues that we could explicitly solve using our biotech technology. And we came across this green seed problem and this sclerotinia problem. And because it's so local, because it's so dear to us, and because it's such a big issue for Canada and the world as a whole, really, we thought it would be worth tackling.
Interesting, thank you.
Sarah, what is the biggest risk to developing the fungicide? I know a little bit about biocides and regulations, and curious why you, as a small group, might try to tackle this big effort to get a fungicide or a biocide approved.
Right. So I guess the biggest thing that's holding us back in terms of the fungicides is that the characterization of thiophorabide as a fungicide is very, very new. The very first paper actually detailing its effects as a fungicide was published just last year in 2019 in March, I believe. So it's a new field of study, and it does require a lot more depth and looking into before it can really be pushed as a commercial product. So we definitely do see that there is potential in it in the future. It's just that we haven't incorporated it into our current business model because it's not feasible at like a shorter term scale. That being said, we do believe based on our results that the fungicide is very, very promising. So in the future, we do hope to partner with a research institute and various other labs to kind of have a collaborative like science effort to characterize the fungicide, characterize its effects on the plant, as well as on, I guess, the general ecosystem before it can really be used and sold as a commercial fungicide.
Great.
Thank you. So I've got two screens here, so I'm looking at your circle flows. So then the pigments would be used in theory to generate the antifungal treatment. What is happening to the pigments today?
So even though the antifungal treatment is like the end goal, there are still other established markets for the pigments. It can be used as a clothing or a food dye. They've also been sold as a part of like supplements. So these, like the chlorophylls pigments can be used for other applications. We just think for the sake of the circularity of the system, in the end, it would be nice if we could repurpose it into an antifungal.
And today, is it currently not, is it a waste byproduct today?
Yeah, so the issue with the current clay system is that it binds the chlorophyll and it actually can't separate from the chlorophyll. So there isn't really anything you can do with the chlorophyll after the fact. But with our system, because our protein can actually release the chlorophyll once it's bound, we actually open up a whole new, I guess, set of uses for that chlorophyll. It just doesn't go straight to waste. We can actually use it for other things. Yeah.
Got it. So today you're able to release the chlorophyll pigments from your protein.
Mm-hmm.
And so then future research is to figure out how can you make use of the chlorophyll instead of it becoming a waste stream. So do you recover that protein? Is that continuously recovered?
Yeah, the protein can be recovered and they can be used up to 7 to 10 times, I believe. Mm-hmm.
Got it. And then what happens?
The proteins are biological molecules, so they can be like broken down really easily and decomposed, composted, and they could be recycled back into the protein production stream as well. So the components that made up the proteins can be broken down and they're fed back into the bacteria, and then they can make more protein.
Thank you.